Hello, I'm Renske de Wit. I'm a PhD student in the group of Sanne Abel and Raymond Feineman. And today I want to talk to you about characterizing the landscape of LINE-1 activity in colorectal cancer. LINE-1 elements are DNA retrotransposons, which can copy and paste themselves to other places in the genome. And they do this using an RNA intermediate. And actually, 17% of our genome consists of copies of LINE-1 elements, but only 100 and 150 remain that are still retrotransposition competent. And normally in healthy somatic tissue, these elements are repressed. But what we observe in cancer is that they frequently become active, and their role in tumor development is not yet understood. So to understand their role in cancer development better, recent work by the PICO Consortium has actually studied thousands of primary tumor tissues and identified 124 recurrently active LINE-1 sources. In this project, we want to follow up on this work and study the presence and the pattern of LINE activity in colorectal cancer, and specifically metastatic colorectal cancer. To do this analysis, we studied a dataset of 610 metastatic colorectal cancer tissues that were made available by the Hardwick Medical Foundation. And these samples had been analyzed by whole genome sequencing and then used as input for the ISMB DNA analysis pipeline, which could identify the line insertions in the tumor genome. And in principle, in our dataset, each retrotransposition event was represented by maximally 4 break ends, and 2 of those were located in the reference genome to the place of the insertion, whereas the 2 others, these 2, corresponded to the location of the LINE-1 source that produced the retrotransposition. And this is also how we distinguished source locations from destinations locations in our dataset. LINE-1 sources are regions in the reference genome that accumulate across our dataset a lot of break-ends, which can be seen here, whereas the destinations are all the break-ends that are scattered throughout the genome. And this is how we separated those 2 in our dataset. From our analysis, we make a number of observations. The first is that almost 99% of our samples display at least 1 retrotransposition event. So it's really common. But what we do observe is that there is a large variety in the activity across the samples. So there are a few samples that only have 1 or 2 events in total, whereas there are also samples that can literally have hundreds of retrotransposition events. If we study the insertions that we detect in detail, we find that the majority of our events are not traceable to the original source. And this is because line sources have very high sequence similarity between them. However, for the events that we get that we do trace back, we find that in our dataset at least 19 recurrently active sources. One of those sources is active in almost 17% of our samples, whereas other sources are active in only a few samples. So there's also a variation in this respect. And we also see that individual sources differ in the number of retrotranspositions that they generate in a single sample. If we study all the locations that the line insertions occur, we find that 1/3 approximately coincides with a gene. And in our dataset, PTPRD was the gene that was most frequently affected, like in most samples, namely 57. And in total, we identified 33 genes with at least 1 line insertion in at least 5% of our samples. This brings me to the conclusions. So in this project, we study line activity in metastatic colorectal cancer. We find that in our dataset of over 600 samples, nearly all samples display at least one retrotransposition event. We see that there is variety in the number of retrotranspositions per sample, which can range from a few to hundreds of events. These events are caused by at least 19 recurrently active LINE-1 sources. And if we study these sources in detail, we find that they vary in the number of samples in which they're active, and also if they're active, the number of events that they are responsible for in a single sample. And we also find that a subset of the insertion coincides with a gene or an exon. In the future, we plan to study line activity in earlier stages of CRC, and we will do that in collaboration with Sargentis, which is a company which has developed a specific technique called FFPE TLC to hopefully study these line events in even more detail. And what we also plan to do is to refine the definition of variant allele frequency and optimize it for line events to better reflect the timing of particular retrotransposition events during colorectal cancer development. And with that, I arrive at the end of this presentation, and I thank you for your attention.